The aim of the lectureship, which is generally sponsored by Sanofi, is to recognize an individual's innovative leadership and outstanding contribution to the advancement of knowledge in the field of diabetes and related metabolic conditions. The 52nd Claude Bernard Medal goes this year to Professor Takashi Kadawaki. It is a great pleasure for me to present this medal to Professor Kadawaki, who is currently the president of Toranoma Hospital. He serves as a chairman of the board of directors of the Japan Diabetes Society and chairman of the boards of directors of the Japan Society of the Study of Obesity, and also serves as vice president of the Japanese Association of Medical Sciences. Professor Kadawaki has made seminal contribution to our understanding of many aspects of diabetes, in particular the genetic of diabetes, the structure and the function of the insulin receptor and the implication in terms of the insulin resistance, the incredible work done on adiponectin. So this has been always, seminal results. He has been also serving in many functions to the community because he has been the associate editor of Diabetologia. He has been working with Diabetes Care in the editorial board of Diabetes of the Journal of Clinical Endocrinology and Metabolism, and is currently in the editorial board of Cell Metabolism Molecular Mechanism. Thus, Professor Takashi Kadawaki has shown innovative leadership and made outstanding lifetime achievement in diabetes research. And today, Professor Kadawaki will deliver the 52nd Claude Bernard Lecture entitled, What is Type 2 Diabetes? A Long Journey to Seek the Truth. Professor Kadawaki, let me just join with the rest of the membership and the community of diabetes to congratulate you and to thank you for the incredible work that you have done throughout your career.
First, I'd like to thank the European Association for the Study of Diabetes, especially Professor Stefano Del Prato, the president of the EASD, for honoring me with the Claude Bernard Award, the EASD's highest award. This morning, I am going to deliver the 52nd Claude Bernard Lecture entitled What is Type 2 Diabetes? A Long Journey to Seek the Truth. This is my COI disclosure. This is an outline of my talk. Firstly, insulin resistance versus insulin secretory defect in the pathogenesis of type 2 diabetes. Second, the role of ILS1, ILS2 in obesity-linked insulin resistance and type 2 diabetes. Third, the role of adiponectin receptor in obesity-linked insulin resistance and type 2 diabetes. Finally, genetic basis of susceptibility to type 2 diabetes in Japanese and East Asian populations. I would like to begin by paying tribute to my first mentors in diabetes research. In particular, Professor Kinori Kosaka taught me to think about what is diabetes day and night. My first project was to determine risk factors for the development of type 2 diabetes in subjects with impaired glucose tolerance. Using proportional hazard model analysis, I demonstrated that a decreased early insulin response to glucose, obesity-induced insulin resistance, and a family history of diabetes were independent risk factors for type 2 diabetes. With these three fundamentals becoming the bedrock upon which my research was based over the next 35 years. Indeed, in the past 35 years, there has been an epidemic of diabetes in Japan and worldwide as well, which is most likely the result of a complex interplay between genetic factors such as reduced insulin secretion and the environmental factors such as high-fat diet-induced obesity, visceral fat accumulation, and consequent insulin resistance. The first part of my presentation will focus on insulin resistance versus insulin security defect in the pathogenesis of type 2 diabetes. Dr. Masato Kasuga taught me the fundamentals of research practice, including how to use a pipette. He always stressed the need to keep an open mind when analyzing experimental data. My mentor in the USA is Dr. Simeon Taylor. Simeon always told me, be nice and fair to your colleagues and especially competitors in science. I am very grateful to Simeon and Dr. J.C. Ross, branch chief, and colleagues, including Hiroko, my wife. We worked very hard. With Dr. Simeon Taylor, I was very fortunate to identify the first insulin receptor gene mutations in a patient with extreme insulin resistance, which served as the proof of concept that this protein mediates the insulin action in humans. A companion paper by Steiner and Bell co-reported an insulin receptor gene mutation. We identified a number of mutations of the insulin receptor and classified them into 5 functional classes according to the life cycle of the insulin receptor. One of these was a mutation of the tyrosine kinase domain, which served as a proof of concept that tyrosine kinase is crucial for glucose homeostasis in humans in vivo. In November 1988, I presented a Nature paper to the NIH Journal Club reporting the invention of knockout mouse technology by Mario Capecchi. I was enormously impressed by this paper, and my colleague Mimo Achiri said he would generate insulin receptor knockout mice, which he did. I said I would generate IRS1 knockout mice. After I returned to Japan, I was fortunate to generate IRS1 knockout mice, which were indeed insulin resistant, but maintained a normal glucose tolerance due to compensatory hyperinsulinemia. This work was the first to demonstrate that IRS1 is indeed involved physiologically in insulin action in vivo. Unexpectedly, however, IRS1 knockout mice failed to develop diabetes, demonstrating that insulin resistance by itself may not be sufficient to cause diabetes. A companion paper by Ron Kahn and associates showed similar results. IRS1 knockout mice showed mild insulin resistance as compared with insulin receptor mutations, suggesting that both IRS1 dependent and IRS1 independent signaling pathway may exist for insulin. Dr. Tobe identified a 190 kilodalton protein, which is tyrosine phosphorylated by insulin in the liver of IRS1 knockout mice, which was subsequently referred to as IRS2. We also generated IRS2 knockout mice. Although IRS2 knockout mice showed similar degree of insulin resistance to IRS1 knockout mice, IRS2 knockout mice developed frank diabetes, which confirmed the results of Dr. Maurice White's laboratory. When we examined pancreatic beta cells, IRS1 knockout mice showed compensatory beta cell hyperplasia to insulin resistance and maintained normal glucose tolerance. In contrast, IRS2 knockout mice showed a defect in compensatory beta cell hyperplasia to insulin resistance and developed diabetes. We also generated beta cell specific heterozygous glucokinase knockout mice, which showed a decreased insulin response to glucose and impaired glucose tolerance but not frank diabetes. This data shows the results of blood glucose and insulin during IPGTT in wild-type mice in blue, IRS1 knockout mice in orange, and beta cell-specific glucokinase knockout mice in purple. We then crossed these mice to generate double knockout mice in red, which developed frank diabetes. Thus, the genetic abnormalities, each of which is non-diabetogenic by itself, cause overt diabetes if they coexist. IRS1 knockout mice and heterozygous glucokinase knockout mice developed diabetes because the individual beta cell function is defective in regard to insulin secretion, which is unable to compensate for insulin resistance despite compensatory hyperplasia. Thus, defects in the compensatory response to beta cells, either hyperplasia or hypersecretion to insulin resistance, played a crucial role in the development of type 2 diabetes. I will now focus on the role of IRS1, IRS2 in obesity linked to insulin resistance and type 2 diabetes. Insulin signals are mediated via IRS1 and IRS2. The liver possesses both IRS1 and IRS2, whereas other tissues express either IRS1 or IRS2 predominantly. My laboratory, as well as Dr. Morris White's laboratory, generated tissue-specific IRS1 or IRS2 knockout mice, which were used to elucidate various roles of IRS1 and IRS2 in glucose homeostasis. This morning, I'm going to talk about the role of IRS2 in macrophages. The hallmark of obesity is macrophage infiltration and chronic low-grade inflammation in white adipose tissue and the liver, accompanied by upregulation of inflammatory cytokines and downregulation of anti-inflammatory adiponectin, leading to insulin resistance and atherosclerosis. In macrophages from adipose tissue of high-fat diet-fed obese mice and OB mice, expression of IRS2 are markedly reduced, and IRS1 was hardly expressed in macrophages. This shows the mechanism by which IRS2 is downregulated in obesity. IRS2 expression is driven by FOXO. Phosphorylation of FOXO by insulin after eating or in obesity promotes its nuclear exclusion, thereby downregulating IRS2. IRS1 does not undergo such downregulation by insulin. In order to determine the role of downregulation of IRS2 in macrophages, we generated mildly specific IRS2 knockout mice. Under high-fat diet, MIRS2 knockout mice showed glucose intolerance due to insulin resistance. Insulin-stimulated phosphorylation of AKT was decreased in the liver and white adipose tissue of MIRS2 knockout mice, but not in the skeletal muscle. Thus, MIRS2 knockout mice showed hepatic and white adipose tissue insulin resistance under high-fat diet. Macrophage M0 acquires the M1 phenotype upon stimulation with interferon gamma and lipopolysaccharide. M2A expressing arginase Fis1 is induced by exposure to IL-4 or IL-13. M2B expressing IL-10 is induced by a combined exposure to immune complex and toll-like receptor IL-1 receptor ligands. M2 macrophage activation is known to be impaired in obesity although the role of it in glucose homeostasis remains poorly understood. MIS2 knockout mice showed significantly increased M1 macrophages and M1 marker genes such as IL-1 beta, as well as significantly decreased M2A macrophages and M2A markers such as arginase 1 and Fis1. In M2A macrophages, we demonstrated that IL-4 binding to IL-4 receptor stimulates STAT6 phosphorylation, leading to STAT6 translocation to the nucleus. IL-4 also stimulates IL-2 phosphorylation, leading to AKT activation and FOXO1 phosphorylation, which destabilizes FOX1 HDAC3 and N-core co-repressor complex, both of which are required for full activation of M2 type macrophages. In obesity, IL-2 is downregulated by hyperinsulinemia, thereby causing impairment of the IL-4 stimulated IL-2 PI3 kinase AKT FOX1 pathway, resulting in stabilization of the co-repressor complex and impaired M2 type macrophage activation, which leads to chronic inflammation and insulin resistance. Very recently, we reviewed postprandial glycemic control through gut-pancreas macrophage axis. When we eat, lipopolysaccharide induced by the changes in gut microbiota and insulin produced by pancreatic beta cell in response to glucose reach and bind to toll-like receptor 4 and the insulin receptors of visceral adipose tissue macrophages, respectively. These stimuli lead to production of IL-10 in an AKT mTORC1 dependent fashion. Then IL-10 from visceral adipose tissue macrophages and insulin from pancreatic beta cells reach the liver and cooperatively suppress expression of gluconeogenic genes, thereby controlling postprandial glycemia. In obesity, however, changes in microbiota by feeding is impaired and insulin resistance is induced in macrophages due to reduction of IL-2. These changes lead to impairment of IL-10 production, resulting in postprandial hyperglycemia. To summarize this section, chronic hyperinsulinemia in obesity decreases expression of IL-2 in M2A, M2B macrophages, leading to impaired IL-4 and insulin signaling in macrophages, respectively, causing obesity-linked inflammation, gluconeogenesis, and insulin resistance. We propose the concept of selective insulin resistance in obesity. Obesity-induced insulin resistance and hyperinsulinemia, and which downregulates IL-2 in target tissues, such as in endothelial cells, leading to reduced glucose uptake by the skeletal muscle. Downregulation of IL-2 in the liver in obesity causes increased glucose output and increased steatosis by exaggerated insulin action due to unaltered IL-1 expression and hyperinsulinemia. This exaggerated insulin action in the liver through the IL-1 is also implicated in liver cancer. As I have just shown, downregulation of IL-2 in macrophages in obesity causes inflammation and insulin resistance. These findings led us to ask, what is the molecular mechanism of obesity-linked insulin resistance? That's why we became interested in adipocytes and adiponectin. I will now turn to the role of adiponectin receptor in obesity-linked insulin resistance and type 2 diabetes. What is adiponectin? Adiponectin is a fat-specific hormone which is the most abundantly expressed adipokine in white adipose tissue. Adiponectin was discovered by 4 independent groups. However, the function of adiponectin was not known. KKAY mice on a high-fat diet become obese and develop insulin resistance, which is associated with reduced plasma adiponectin levels. Importantly, replenishment of adiponectin reverses insulin resistance and metabolic syndrome in KKY mice on a high-fat diet. These data clearly indicate that adiponectin is indeed an insulin-sensitizing adipokine, and also indicate that reduction of plasma adiponectin levels in obesity plays a causal role in obesity-linked insulin resistance and metabolic syndrome. A companion paper by Philip Scherer and associates showed similar results in the liver. We have been proposing adiponectin hypothesis. According to this hypothesis, reduced plasma adiponectin levels caused by genetic and environmental factors such as high-fat diet-induced obesity play a crucial and causal role in the development of insulin resistance, type 2 diabetes, and atherosclerosis, data not shown. In 2003, we cloned adiponectin receptors AdipoR1 and AdipoR2, which are predicted as integral membrane proteins with 7 transmembrane domains, whose N-terminus is internal and C-terminus is external, opposite to GPCRs. AdipoR1 mediates activation of AMP kinase, and sirtuin PDC1 alpha, a master regulator of mitochondrial biogenesis, as well as activation of calcium influx. AdipoR2 mediates activation of PPAR, alpha delta gamma. Using tissue-specific AdipoR1-R2 knockout mice, we determined the role of AdipoR1-R2 in anti-diabetic actions of adiponectin in liver and skeletal muscle. More recently, we have demonstrated anti-inflammatory actions of AdipoR1 in macrophages and the endothelial protective function of AdipoR2 in endothelial cells, both contributing to anti-atherosclerotic actions of adiponectin. The anti-inflammatory action of AdipoR1 in macrophages also contributes to anti-diabetic actions of adiponectin. Decreased adiponectin effects in obesity due to reduced plasma adiponectin secreted from adipose tissue and downregulation of adiponectin receptors in target tissues are causally implicated obesity-linked diseases such as type 2 diabetes, fatty liver, atherosclerosis, and even obesity-linked cancer. Consistent with this, AdipoR1 knockout mice, AdipoR2 knockout mice had a shorter lifespan, and AdipoR1-R2 double receptor knockout mice had the shortest lifespan under high-fat diet. Interestingly, AdipoR1-R2 double receptor knockout mice had increased liver and colon cancer, both known to be obesity-linked cancers. Caloric restriction, exercise, and the adiponectin receptor pathway all activate common sirtuin and AMP kinase pathways, which may increase longevity in humans. we embarked on developing an orally administered adiponectin receptor agonist as a caloric restriction and exercise mimetic agent. We obtained one such adiponectin receptor agonist, referred to adipolone. Administration of adipo-R1-R2 agonist, adipolone, in vivo showed pleiotropic effects in these tissues, such as increased insulin sensitivity, normalization of glucose lipid metabolism, energy dissipation, anti-inflammatory action, endothelial function, anti-cancer, and healthy longevity. In 2015, we solved the 3D structure of human adiponectin receptor. As we predicted in 2003, adiponectin receptor is a 7-transmembrane domain containing receptor whose N-terminus is inside the cell and C-terminus is outside the cell. Remarkably, we found a zinc ion bound within the 7 transmembrane domains in the adipo-R1-R2 structures. This is indeed the first example where a zinc ion is bound within the transmembrane domain of any receptor protein. This slide depicts the coordination of the zinc ion, shown in magenta, by 3 histidine residues and aspartic acid of adipo-R1 and adipo-R2 viewed from the cytoplasmic side. In the adipo-R1-R2, a water molecule shown in red is also coordinated to the zinc ion. An attractive hypothesis is that adipo-R2 might have zinc ion-dependent hydrolytic activity, and free fatty acids might be produced via lipid hydrolysis of the membrane by the adipo-R2 in response to adiponectin, leading to PPAR alpha activation by the produced free fatty acid. This shows the cavity of adipo-R1 and adipo-R2. The cavity extends from the cytoplasmic surface to the middle of the outer lipid layer of the membrane. In a recent study, we determined the crystal structure of adipo-R1, which is fully active with respect to the downstream AMP kinase activation, and found that it represents a dual conformation consisting of the closed form and the open form. These observations indicate that adipo-R1 and adipo-R2 data nets shown appear to assume conformational change between closed and open forms. In the open form structure, the cytoplasmic side of the helix 5 of the adipo-R1 is shifted rightward, thereby markedly increasing the cytoplasmic opening of the major cavity and facilitating signal transduction through adipo-R1, presumably. In fact, we have recently identified a novel zinc-dependent hydrolase activity towards membrane lipids distinct from ceramidase, intrinsic to adipo-R1 and adipo-R2. In summary for this part, we discovered adiponectin receptor adipo-R 17 years ago, developed small molecule compound targeted adipo-R, adipolone, and elucidated 3D structure of adipo-R. We are in the course of optimizing adipolone based on the 3D structure, which may serve as the first-in-class drug for obesity-linked diseases aiming at improving healthy longevity. I would finally like to look at the genetic basis of susceptibility to type 2 diabetes in Japanese and East Asian populations. Asians are susceptible to type 2 diabetes despite a mild degree of obesity due to low insulin secretion, which is genetically determined presumably. until recently, approximately 100 type 2 susceptibility loci have been identified. However, most, if not all, are common across ethnic groups. Therefore, Asian-specific type 2D loci had not been identified. Recently, we carried out a large-scale type 2D GBAS in Japanese subjects using over 12 million variants. This is Manhattan plot. We determined genome-wide significant associations at 88 loci, of which 28 were novel. This is schematic of common and specific type 2D genes in Japanese subjects in the present study and in Europeans. Pathway analysis showed several pathways such as MODY or beta cells were commonly identified in both populations. On the other hand, a fraction of the identified loci, such as variants in GP2, PAX4, and GLP-1 receptor in Japanese, were rare or monomorphic in Europeans, which can lead to differences in the association of pathways in the two populations, as is the case with regulation of insulin secretion in Japanese. In the Japanese and European type 2D GBAS, GLP-1 receptor region showed a significant association with type 2D only in Japanese, while the minor allele frequency of R131Q substituting glutamine for arginine was 18% in Japanese, and it was less than 1% in Europeans. This missense variant in GLP-1 receptor is reported to be associated with greater than twofold increase in GLP-1 stimulated insulin secretion in healthy adult humans. In fact, this missense variant was a protective allele for type 2D. Approximately 1/3 of Japanese subjects are heterozygous or homozygous for this variant, but this is extremely rare in Europeans. Since this variant in heterozygous subjects is associated with increased insulin secretion to exogenous GLP-1, it may serve as a marker for responsiveness to GLP-1 receptor agonists and DPP-4 inhibitors in Japanese and East Asians in whom such incretin-based drugs have been shown to have more potent glucose lowering effect than in Europeans. We then determined the enrichment of heritability in 220 cell types in the type 2D GBAS and observed the most significant enrichment in H3K27 acetocytes, characteristic feature of active enhancers in pancreatic islets. These data strongly suggest that major genetic heritability of type 2D comes from pancreatic islets. This compares to largest GBAS in European and in Japanese, which identified 135 new loci and 28 new loci respectively. Surprisingly, only 8 loci were common, indicating that we have begun to identify ethnic group-specific type 2D loci. Most recently, the largest meta-analysis of type 2D data from East Asians was reported. In fact, we identified 61 loci that are newly implicated in the predisposition to type 2D. These data strongly suggest the genetic susceptibility to type 2 diabetes preserved among East Asians, but not necessarily in other ethnicities, such as Caucasians. Givas to the People is the title of the editorial in the October 2018 issue of Nature Medicine. It says that thanks to improvements in data collection and analysis, polygenic risk scores, PRS, that predict polygenic disease risk are approaching the same predictive accuracy offered by tests for monogenic mutations. The time to think about how best to incorporate polygenic tests in the clinic is now. We therefore try to predict the risk for type 2D in Japanese according to a polygenic risk score. The polygenic risk score for type 2D in Japanese was calculated using 80,000 SNPs. The results are shown here. The odds ratio for top 10 to bottom 10 percentiles was around 5. Importantly, the polygenic risk score for type 2D in European failed to predict type 2D in Japanese. This morning, I am very honored to present the Claude Barnard lecture entitled What is Type 2 Diabetes? A Long Journey to Seek the Truth. I talked about insulin resistance versus insulin security defect in the pathogenesis of type 2 diabetes, the role of IRS1, IRS2 in obesity linked to insulin resistance and type 2 diabetes, the role of adiponectin receptor in obesity linked insulin resistance and type 2 diabetes, and finally, genetic basis of susceptibility to type 2 diabetes in the Japanese and East Asian populations. While I look forward to continuing my research journey, I wish the younger generation of researchers to take up the challenge to gain further insights into the disease. I would like to acknowledge my mentors, in particular Masato Kasuga, for his long-standing mentorship. I am extremely grateful to Professor Toshimasa Yamauchi for his enormous contribution to the work I have been presenting this morning. I also acknowledge my colleagues, Naoto Kubota and Koji Ueki, for their excellent work. I am very grateful to all the former and current members of our laboratory, shown and not shown in this slide. I am very grateful to my secretary, Kazuko Nita, who has been working with me for more than 20 years. I could not have achieved this much without her excellent support. Lastly, and most importantly, I am extremely grateful to my family. I could not have done my work without them. Thank you very much for your kind attention.
Professor Kadohashi, thank you so much for this enlightening presentation. I mean, this has really been taking us through an incredible journey of what we know and we still need to know about type 2 diabetes. Again, let me conclude and congratulate you for this 52nd Claude Barnard lecture.